yeah, it works. So, good afternoon, everyone. I'm happy to see some people here, given that this is the very last session of the whole ABRF 2025. Thanks for coming. We will have great talks, I promise you. There is one by Leica and two great scientists, microscopy physicists. But let's say a few words about where I'm coming from. So I'm Radu Moldovan. I'm running the microscopy core at the Anschluss Medical Campus in Colorado. We have a great arsenal of instruments. We started in 2010 with trying to get our very first Leica STED, which wasn't that great, but 15 years later now, I'm pretty sure we'll be all agreeing that their new STED system is amazing. And Haridas here, the product manager, confocal product manager on North America, will talk about that. After that, we'll have Dominik Stich, a physicist in our core, talking about STED. And I assume we all know about STED. So he will talk about the challenges of doing STED with users on a medical campus. We're a medical campus. And then, last but not least, we have Doug Shepard from Arizona State University, associate professor in physics department, talking about his implementation in SIM, structural illumination microscopy. So let's start with Haridas. Thank you.
Thanks, Radu. Welcome to the session. So I hope it's a kind of a short session, but I will try to see what I can convey here in 10 minutes about STED, right? So obviously, STED technique is a complex system, but our goal also from Leica's side is to simplify it for the user. That is so tau STED, tau STED extend. Tau STED extend is the new iteration of our STED system. So as Radu already mentioned that 15 years back, right, we had a CW STED, then gated STEDs. There's a whole transition. Rich also is very familiar with that history. So, hope it moves forward.
It's not plugged into your computer. You have to press on your computer.
No, this is mine. Okay, well, whatever reasons. Happened. Okay. So what do these all images have in common? Right? All of them happens to be STED, live cell STED images. Live actin amnion green, mito orange, live actin YFP. So you can see traditionally, historically, with STED and live cell, there was a kind of almost like, okay, it's too much power. You cannot do it. Was the concept, right? But over a period, that has significantly changed. That is the key message. Just I can stop this slide and go home, right? You can do super resolution STED image with the live samples, right? So, and that is not just 2D in 3 dimension, and multicolor, all of them possible, right? So you can see that mitochondrial crystal, live cell imaging, right? So what is it? The Stellaris confocal platform is what is making it possible, but history, exactly the way it was set up was perfect, because the story started in 2004 with the 4-pi system. Many of them came from Stefan Hell's lab. And then over a period, there's a whole range of changes, evolution of the system, right? Right now, the latest one iteration is tau STED extend. So proportionally, the depletion power, laser power needed for imaging was continuously going down. That was the whole goal, keeping the resolution similar, right? And this was the original. So this guy here is Stefan Hell's postdoc, graduate student that time, the first paper, STED paper, Marcus Daiba. This was the setup which we converted to this package, right? So you can imagine that complexity of a system, how the conversion happens. And so, oops, moving too fast. So this again here, originally, it's not playing automatically. Yeah, you can see. So the original STED system was purely relying on power, increasing power to increase resolution. But later, we moved on to adding the lifetime domain. A phasor analysis, fluorescence lifetime, center of the donut having a longer lifetime, periphery having shorter lifetime. Using phasor unmixing, you can actually extract the information. So that making use of that lifetime actually played a significant role in decreasing the depletion power already. That we named as tau STED. So the next iteration is tau STED extend, extending further. That was the. So if you just look at the standard nuclear pore sample, confocal, a regular time gated, again lifetime using gated STED image, tau STED extend. Right? If you use this, so the point is that you can use a 0.5%. Again, it's a relative number, but still, typically we used to end up using like 20%, 30%, 40%, certain wavelength, even 50, 60% power. Now in a 0.5% power, you can get this kind of an image. So that iterative progress in maintaining resolution with a lower and lower power, laser power. That is the core message I wanted to convey here. Because many of you, most of you are really familiar with STED, what it can do. Here the main goal is to make it compatible for live cell imaging. Right? So this again, you see that 75 nanometer resolution, that is not the best resolution obtained in this sample, but this was at a 0.5% power. If you increase 1%, 2%, okay, you can get back to, but again, nuclear pore size is, you cannot get 25 nanometer because the pore size, that antibody size, that itself is much bigger. Right? So, again, establishing that resolution, so in the end, that live cell imaging is the ultimate goal at that high resolution. This is again 99 frames at a 2.5% laser power, depletion power. So this is made possible by array of technologies from Leica. The concept of STED came from Professor Hell's lab, but there were some added things actually coming from Leica in the sense that the photon counting detector, hybrid detectors, built-in film, photon counting electronics, spectral confocal, right? All those, the spectrometer, built-in spectrometer allows you to adjust it, white light laser, all those components plays a role. It all adds to the technology to make it possible. And this is another example of life-acting neon green. So you can see 30 minutes STED imaging at the highest resolution possible. Right? Another one, 20 minutes, 600 frames. Right? Highest times, temporal and spatial resolution possible. And still cells are active and live. You are not killing the cells. So that's at the core message I wanted to convey in this such a short window. There is a lot more to talk about it, but
yes, if you have questions.
So are you still seeing a linearity between power and resolution, even though you've added these other components?
yes. So I don't know whether it can call linearity because the lifetime is also the change in lifetime is proportional to change in depletion. If you are able to, so 50% depletion introduced, proportional change in lifetime will happen, right? Because, but at the same time, I don't know whether you can call it linear dependence on power, because the photophysics, how exactly it will evolve in terms of lifetime, the relationship.
Well, because now you're playing two things. Right?
Two things. Yeah. But there is a dependence on power. So you can keep the power extremely low and keep X resolution, or you can increase the power and further improve the resolution as well, right? But depends on the biology, and you can play with it pretty much.
I do have a question.
Yeah.
So, what's the dwell time per pixel now with this very low stat? I mean, is it something?
So if you think about that 600 frame in 20 minutes, right?
Yeah.
That will translate into around 20 frame per minute kind of a translation, right? So, and at the STED resolution, you cannot undersample. You will have to oversample pretty much. So if you calculate it back, right, dwell time is relatively small.
So it's,
If the signal is good, again, it's all signal dependent. If signal is good, so at MBL course actually recently, I was using resonance scanner for say mitochondrial crystal imaging. So usually that's a 25 nanosecond dwell time, one pass, but you may have to average some, but, or accumulate or average, right? You have mostly accumulate here. But even then, right, if you add up also, it will be hundreds of nanoseconds only, right? Dwell time.
okay. So my question was if you have to sacrifice on speed when you go low with a STED, or not much.
So if you are going slow, so that actually you can sometimes even decrease not decrease. So resolution improvement also, right? If you go with a resonance scanner, there is a limit to which resolution possibly you can expect. Even if I wouldn't say 25 nanometer resolution with a resonance scanner. There will be some compromise. But if you go slow, really slow, right? If you're fixed sample, if you're trying to go with the best resolution, then I will go slow. We use the maximum pixel dwell time so that you get enough photons. Then, because the phasor calculation also, more photons you get, more accurate the phasor calculations.
You can also use accumulate rather than add.
Yeah, accumulate. In counting mode, accumulate.
Yes.
Is there any other questions? If not, let's thank Haridas. Thank you, Haridas. Our next speaker is Dominik Stich from Anschluss Medical Campus, our core, and he will talk about the challenges in doing STED on a medical campus. I just want to remind you to turn off your cell phones, and if you need this talk, these slides, it might be available for certain speakers, not for others, but yeah. Okay, Dominik, please.
Okay. So, as Radu said, my name is Dominik Stich. I've now been with the core facility at the Anschluss Medical Campus in Aurora, Colorado for nearly 9 years. And I was originally hired for doing a super resolution microscopy, as they realized that having a dedicated person for this special set of techniques was really necessary to have any relevant outcome. In my talk today, I will first recap pretty much all of the STED basics, since that is what plays a lot into the problems or the challenges that our users see when they do STED. So, just from the very beginning, STED microscopy stands for Stimulated Emission Depletion Microscopy. It was invented by Stefan Hell and Jan Wichmann in 1994. And then, more importantly, after the invention, it was also demonstrated by Stefan Hell and then Thomas Klar in 1999. So not only did they have the idea, they were able to show that it works, unlike some Russian predecessors who in the Cold War had the idea for STED already. STED is a point scanning super resolution technique, and as my former colleague always called it, it's just confocal on steroids. It allows you to do multi-channel fluorescence imaging. In the lateral direction, you can get down to 30 nanometers. In the axial direction, you can get down to something like 80 nanometers. And so if you do that right, you get these stunning images here of cultured hippocampal neurons where you see a stark contrast in this 2D STED image between confocal and STED. Or if you do 3D, here is like a T cell where the CD122 is labeled. Yeah. So what's actually then the physics background? So the most important part about STED microscopy is the stimulated emission. Now, most biologists are very aware of how to do fluorescence microscopy. And in fluorescence microscopy, you usually use photons to excite a fluorophore. The photon gets absorbed by the fluorophore, and then in normal fluorescence microscopy, you have spontaneous emission, meaning an excited fluorophore randomly gives off a photon and goes back into the ground state. This is just a normal fluorescence that we use. Everyone has seen then these graphs that we use then to characterize the absorption and the emission. And if we then also start measuring the time between an absorption and an emission event and do that over and over and over again, you will get a histogram that is a representative of the lifetime, which is often characterized by a single mono-exponential decay. Now, in addition to this spontaneous emission, you can also have stimulated emission, meaning you have an excited fluorophore, and now you send in a photon. That photon interacts with the fluorophore, and the fluorophore sends out a photon of exactly the same wavelength, direction, and phase. And by doing that, the fluorophore goes into the ground state. While this might sound a little bit esoterical, it actually was proven quite some time ago in 1916 by Albert Einstein. Sorry, not proven, it was hypothesized. And this principle has been widely used in lasers, for example. Now, in stimulated emission depletion microscopy, how does this compare to confocal? So let's just look at the sample. Here we just have a mock-up structure of some linear things. Sorry, I'm not a biologist. Probably some like actin decorated with a few fluorophores. If you now just generate a grid on them, use a focus spot that you focus down as tightly as possible to excite the fluorophores in there. Then you collect these the photons from these fluorophores when they emit the light. You will get a confocal image. In STED microscopy, you still do all the steps that you do in confocal microscopy as well. You just overlay this excitation volume with this kind of donut structure, so that all the fluorophores that were initially excited by the excitation laser now get de-excited under the donut, and only right in the center of the donut, where there's no STED power, does the fluorescence excitation survive. So that means all the light that you're collecting, you're now collecting from the center of this donut. Of course, now that the spot from where you collect is significantly smaller, you also have to make your sampling grid significantly smaller, which usually means 2 more measurement points for the same area. And unfortunately, in 2D it scales quadratically. In 3D it goes with, it goes cubically. And when you then collect the fluorescence, you get significantly better resolved images. In the normal setup of a microscope, you still have your excitation laser. Instead, the excitation is usually pulsed. The STED is also pulsed. Just that way you have a higher STED efficiency. The the pulsed excitation laser, just as normal, excites the sample. The light on the path gets collected through a pinhole, just like in a normal confocal microscope. Now the only thing that you add is your STED laser. You have something that's called a phase mask to then generate this donut profile, and you just couple it in with a normal dichroic mirror into the beam path. When we now have a look at the absorption and emission spectrum, you still have your laser line that excites your somewhere in your absorption maximum. You have your emission bandpass to collect the fluorescence, but now you also have the stimulated emission depletion laser that has to hit the spectrum of the fluorescence emission somewhere where there's still a noticeable fluorescence emission signal. Otherwise, STED simply wouldn't work. And then when we come to generating these donut or like three-dimensional shaped structures, for that we have a phase mask that actually we have two phase masks, one for a 2D STED, one for a 3D STED. Those get overlaid with a laser, and all these phase masks do is they just slightly delay the wave front of the laser. In the 2D mask, it's actually a helical thing, so it goes from 0 to 2 pi when you run around the circle here, meaning that right in the center here, all of your phases are present, which means that you completely simply have no light there, since it's destructive interference. In the 3D phase mask, you have just a center which is at pi, and the outside which is at 0. These phase masks are either printed on glass or are applied using a spatial light modulator. And once you then focus down the laser beam, you either get the donut profile with kind of the torus here for the 2D STED. So this is the lateral profile, this is the profile in the axial direction, where you then see, well, right here in the center, you simply have no laser light from the STED, so you don't have any de-excitation, and you get a really long kind of cigar-like space in here. Whereas in the 3D phase mask, you get the structure which also has kind of a donut in the center, but then most of the energy is actually being deposited right above and below of the excitation volume. When you then look at, well, how does a small bead look? You see, okay, this here is confocal, this here is 100% 2D STED power, where you get this cigar-like shape in the axial direction, whereas in the lateral direction, like in the imaging plane, it's nicely confined. Over here is the pure 3D image, which is isotropical. It's actually even slightly better in Z direction than it is in X and Y, which is kind of freaky for me as a microscopist, because I'm always kind of used to seeing quite some aspect ratio in the resolution. And then the cool part is that you can actually freely tune between 2D and 3D. So you can go from 900% 2D STED power to 95% 2D STED, 5% 3D STED. You already see there is already quite something cut off. And technically, the Z resolution of your 2D is actually worse, or slightly worse than your confocal. And that can go all the way then down to 100% full 3D STED power. Then, when it comes to the resolution, so what I showed you here is just we applied one fixed amount of STED power, and then we see what happens when we shift between shifting the STED power into 2D or 3D mode. Now the resolution in general simply only depends on how much STED power you have available, with the little caveat that your fluorophore has to survive that. Of course, as soon as the fluorophore is bleached, you're done with it. But technically, if you have an infinitely good fluorophore and you have an infinite amount of STED power, you can just go to arbitrarily good resolution. which has been shown in very esoteric materials like diamonds with NV vacancies. But it's not applicable to really biological materials. What is then also important to keep in mind, so this is actually just this formula plotted out, is that different fluorophores have different STED efficiencies. So even though we use the same relative amount of STED power, we will get different resolutions out of it. And of course, in order to get any good STED image, you really need a photostable fluorophore. If you don't have that, you will barely get any resolution improvement in the classical STED. So that then also leads me to, well, what are the most common methods of improving the resolution and reducing the photo bleaching? Well, one idea is to use adaptive illumination. In adaptive illumination, you, for example, first take a confocal image of your sample, and from that image figure out where you have sample and where you don't. And you only turn then the STED laser on in places where you really have some structure that you need to image. That is especially helpful if you go to three-dimensional imaging, since many of the lower laying layers that way get significantly less STED power. And then another idea is, well, if you have a certain pixel dwell time, and what you can do for the first 10th of the pixel dwell time, you count how many photons you have. And if that is above a certain threshold, you keep exposing for the remaining pixel dwell time. If not, you simply turn the STED laser off. That way you also deposit a significantly lower STED and excitation power into your sample, thus reducing the photo bleaching. And then another way of increasing the resolution and also decreasing the photo bleaching is by using fluorescence lifetime imaging, as we just saw in the talk by Laika. So the basic idea, just to go into the detail about that, is that sure, we have our confocal excitation volume again. If it's just a normal confocal excitation volume, we have our mono-exponential decay. If we now overlay that again with our STED laser, the center of from the center of the donut, the fluorescence will not be changed. You just will then detect exactly the same decay. However, all the fluorophores underneath the STED donut either get STEDed immediately, or so that they don't contribute any photons, or if they are able to send out a normal photon before they get STEDed, then the whole lifetime is skewed to shorter lifetimes. So instead of the normal mono-exponential decay, you now get something in the easiest possible view, which is bi-exponential. So you get photons from the center, and the red ones are the photons which are from underneath the STED laser. This is a bio-exponential decay. You can actually separate both of the components apart. And that way you get a short lifetime, a short component and a long-lived component. And if you look at that in images, like this is just a normal intensity. This is an image of beads where we see the slow lifetime in green, where the fluorescence lifetime was not altered. The fast lifetime is where the fluorescence was altered through the STED profile. As you can see, the green is way more confined than the cyan here, and here are then the split up images, which are unfortunately not that well visible. Okay, so these are the basics that are really important to keep in mind when we then start training our users on our systems. So first off, we actually have a commercial system, and there are, to my knowledge, a total of 4 different manufacturers out there that provide commercial STED systems in no particular order. It's ISS, PicoQuant, Leica, and Aberia. And in our light microscopy facility, going into the history of how we got our system, or got started with STED, the first STED system was actually a home-built system, so that's the system I was hired to run, which was borderline fun, because for every imaging session you needed between 30 and 90 minutes just to get it aligned. It was a stage scanning system. You only had 20 micrometers of scan range, but it actually delivered surprisingly good resolution. We had definitely 50 nanometers in the far red channel, something like 65 nanometers in the red channel. But then in 2022, we got the first commercial system, which was an Aberia Steadicon. And then in 2023, we got the Aberia Infinity line that we were able to purchase together with a faculty member and then house in the light microscopy core. So just why do you actually need two systems, or why is it nice to have two separate systems? Well, the Steadicon is only a 2D STED. You can do 2D STED in up to 3 channels. It's also very commonly used as a very good, very sensitive 4-channel confocal imaging, and the reason why it's popular is because it's super easy to use. But then we also have the fully decked out 3D STED, where you can really do everything, anything from 2D imaging, 3D STED, and it also has live cell capabilities. So now we have light microscopy core with one defunct system, and then 2 systems which are somewhat competing with each other, but we actually see them as kind of complementary actually. And so how do we then go about and start training users on the system? Well, first off is that if someone wants to do STED imaging, we usually start with an initial consultation. And I used to joke that I sit them down for the STED talk, which means that first off, I want to understand what is the biological question that they want to answer. And that is simply just to inform myself. I have no biology background to understand what they want to do in their imaging experiment. And that even goes as far as to how should, or what do you expect to see? Like, what kind of imaging data would you hope to get out of it so that you can answer your questions? The next step, and this is really crucial, is to discuss the sample preparation. Then we will plan the experiment, and ideally I also learn about what the trainee's background in microscopy already is. Plus then there's also some questions about how does that trainee usually prefer learning, like is it by me providing additional material, or is it a person that ideally learns by doing themselves? Do they need to write everything down? Do they want to just talk about it, just to cover the basic types of learning? Then, from this initial consultation, we decide if we need to go the route where we go to the simpler system, where we only can do 2D STED imaging, or if we go to the fully complex system. And if we go the route where we go to the easy system, it's a training session consists of first talking about the confocal and the STED theory. So pretty much like a little bit longer version of what I showed you in the first few slides, just so that they get an idea of what needs to be happening in their sample so that they could get good resolution. And then after that, the trainee drives, and we insist that the trainee brings their own samples. And that is a really crucial point, because the trainee is more engaged when they see biology popping up in their sample. Sure, we would have test samples, but that way I kind of ensure that the trainee is engaged. Secondly, if they have already somewhat okay samples, and we try to make sure by actually first putting the samples on an either confocal or an epifluorescence microscope, and if under a high magnification objective the samples don't look good, then we simply don't start the training and everything goes back to the workbench. If we then have a good sample, we in this training session are already generating a set of imaging parameters that the student will be able to use in the future. Then the third reason is by simply preparing the sample, the trainee actually is already committed to the experiment and to the training session, because our facility offers training at zero cost, like it's free for any user on campus, and we simply do not want that to be abused, that some people just wander through the door and it's like, hey, can you show me this for 4 hours? And yeah, that would be a waste of everyone's time. And yeah, then after this training session, we offer continued support. So because once they have the data, the next question becomes, what do I do with this data? So we then throughout their imaging sessions will talk about possibly optimizing the workflow, the settings, but then also talk about data analysis and hopefully plan future experiments where we then might also use the more complex system. In the more complex 3D STED, we do the same thing where we require the user to bring a sample, but there the trainee doesn't drive the system right away. The system is so complex and overwhelming that unless it's a super familiar user with STED, in our experience it's best that in the first training session the ALMC personnel shows the user how to run an experiment and talk about all the different features that we had just talked about in theory on a sheet of paper, ideally on their sample. And then once we get through that session and then a few days later have the second session, the trainee fully takes over and then hopefully has retained some things of the first session, which is unfortunately not always the case, but very often it is. And then in the end, there's still all the continued support that we offer. One thing I want to just briefly go into detail about is the importance of sample preparation. So in STED microscopy, you always, with the current systems, want to use the far-red channel for the feature that you're most interested in your sample, simply because the fluorophores are the most photostable and they offer you the best resolution. The second channel to use is the normal red channel, which uses exactly the same STED laser. Since the same STED laser has less of an overlap with the fluorescence out there, the STED capability is not quite as good, but you still get quite brilliant resolution. You just will have to remember to crank the STED power up a little bit more if you want to get similar resolution as in the far-red channel. A normal microscopist would be like, okay, I need a third channel, let's do it in the green. Well, that is a horrible idea, because if you then see where this would be steaded, you notice that the STED laser is nicely overlapping with the the absorption spectra of your red and far-red fluorophore. So as soon as you turn the STED laser for the green channel on, you very effectively bleach the heck out of your sample. There is nothing left. You can do it, but then you have to do it in a separate step and first finish all of your imaging in the far-red and in the red channel. Also, if you do the green channel imaging in an extra step, you lose one of the really cool built-in features of a STED, and that is that all the channels are perfectly aligned. You barely have any other system where that is the case. Since the STED laser confines or like turns off all the fluorophores in the periphery, and the fluorophores can only fluoresce right in the center of the donut, all of your channels that you image with the same STED laser are perfectly aligned. That is definitely not the case in any confocal microscope. But yeah, instead of using a green fluorophore, it's often better to just use a long Stokes shift fluorophore, which you excite here in the blue range, but yeah, the fluorophore then emits in the red, and you still can use the far red STED laser. So that way we already have 3 STED channels. And if that is not enough for you, you can think about loading up fluorescence channels with 2 or even 3 fluorophores. As long as they have a significantly different lifetime, you're able to tell that apart just using fluorescence lifetime imaging, just as we improved the resolution on the normal one-channel experiment with using the fluorescence lifetime imaging. And yeah, if that is not for you, you still can think about adding a green fluorophore to it if you have a STED laser for the green. And doing that kind of scheme, both Aberia and Leica, to my knowledge, demonstrated that you can have up to 6 STED channels. Okay, sample preparation. Going a little bit further, so sample preparation is also a really good option for you to minimize aberrations in your sample. And the best way to minimize aberrations is by, first of course, using the correct cover slip. And then you can choose your mounting medium so that you increase the transparency of your sample. And you can also change the refractive index of your sample to something that is really close to the refractive index of oil, so that you minimize the spherical aberrations. We have some good experiences with different kind of mounting media. Prolong works great. Moviol is really great for 2D STED. The TDE mounting medium is interesting because you can make it pretty much for dirt cheap, but you can adjust the refractive index, and it has a slight clearing capability. And we have also used slow fade glass to get a stronger clearing effect of many of our sliced tissue. And then, yeah, sample preparation is also a good way to reduce your background. So you want to avoid DAPI or Höchst in the mounting medium, meaning like the mounting media that already come preloaded with it, don't use it, because that will get excited in these high concentrations and it's omnipresent in your sample. If you need it, do it in an extra labeling step. And then you have a sample, and you might find yourself lost when you use the 3D STED in the resolution space that the STED offers you. On the one hand, you know that the more power I put in, the better the resolution becomes. I can then also start tweaking from 2D to 3D. And the really tricky part then is, how can you actually make sure that you get the resolution that you need? And to this point, there is not a good answer for that. In confocal microscopy, we're used to being able to calculate the resolution that we have in X, Y, and Z. Whereas instead, there is a formula that I showed you. It's different for every fluorophore. It's massively fluorophore dependent, it's environment dependent, it's sample dependent, what kind of resolution you get out of it. And yeah, there's unfortunately also no standardized way of really figuring out what the resolution in your sample is. When you then, for example, talk to some manufacturers and suggest ways of what they could implement, one of the answer is, well, if we implement a resolution measurement into our system, we will be held accountable to that. And so they do not give us this special tool as we're imaging, which would be tremendously helpful. And, oh yeah, I already mentioned that each imaging channel is different. But yeah, then here, for example, we have an experiment where we look at cultured neurons again. And yeah, there you see these 3 channels definitely have a different resolution. As long as you only want to get crisp images, that is not a problem. These images are still so much better than any confocal image. However, it becomes really tricky if you start to do some advanced analysis on this. For example, if you think about the cluster size. So when you do this analysis, you simply do not know what is the smallest detectable volume or area that you can get out of it. So you do not know your lower threshold or your lowest sensitivity in the system. And that, to this point, is a big problem in some experiments, unfortunately in many of the experiments that we run. We're still trying to figure out workflows and working on calculating resolution not only in 2D, as you can do with Fourier ring correlation, but also in 3D. And yeah, if you then have done everything right, you, as I mentioned, can get very brilliant images. Here's just the example from the beginning, where we show cultured neurons, confocal versus STED. Here is some microvilli that a researcher imaged. Of course, if you go down further and further, you might find that the underlying structure is very punctate, and that can be very disorienting as well. So it might not even be necessary to crank the microscope up to 11, but only measure at a moderate 4 or 5. Then there's every now and then a sample where the researcher is like, I have no clue what these red blobs are, and but they're really pretty, and I'm just like, yeah, it's a good background picture with no specific biological value. Then, yeah, other samples that we have imaged are, for example, tetrahymena. There in the large overview you don't see it. This is confocal. This is STED, where you then really see the individual structures. And then as soon as you go to 3D, yeah, imaging speed becomes a real consideration. Oh, I should have not done it this way. Yeah, this way both start. And yeah, in this case, the researcher really realized that we do not crank up the resolution massively, just well enough so that we can clearly see the branching points and how well the mitochondria touch. And yeah, then this here is just, again, one of the images from the beginning, like the T cell. And yeah, that would be it for this presentation. Thank you very much.
Dominik. Thank you. We have time for a very short question. If you don't have a question, we do have a question. Okay, very short question while Doug is setting up.
Okay, so I didn't quite get, why can't you use the kind of conventional methods to figure out what your resolution is? Is it because the sample responds differently to the depletion beam, or why can't that be measured as in more conventional techniques?
Well, which techniques do you mean?
Well, like confocal or anything like
Well, in confocal, the resolution measurement is simply a theoretical determination.
But you can also measure with a bead.
Yes, you can measure with a bead, but none of the manufacturers tells us which fluorophore they have in the bead.
Okay.
And since then all fluorophores stat differently, it's an idea of what you can get as a resolution, but it's not necessarily a trustworthy measurement in our experience.
So it's not good enough for whoever
So,
bead manufacturer to tell you that it's got the spectra. You actually have to
it's not only the spectra, it's really the photophysics of the fluorophore.
Gotcha. Okay.
So different fluorophores have a stat differently well. And so different, or the same excitation power on two different fluorophores in the same spectral range will likely give two completely different outcomes.
All right, let's start the last talk. Doc Shepard, he will talk about structural illumination implement.
Great, so thank you. The resolution is not going to be quite as good here. So I'm just curious, how many people have done structural illumination? How many people have had problems with artifacts in the structural illumination? All right, good, this talk's for you. So, my lab, we are a biophysics lab. I have a long history on working on optical methods development. I actually started my lab at the university where Radu and Dominik are, and then moved it to Arizona State about 5 or 6 years ago. So we are physicists, but we're about half biologists, half physicists. And recently we've been working a lot on structural illumination because it's a method similar to STED in that it's really driven by the physics of the microscope, but it's a little different in that we don't have to worry about the fluorophore physics that was just talked about quite as much. There's a nonlinear version of this. I'm not going to talk about that today. So what I have here is some data from a colleague of mine in France, Christophe Lettier, where he gladly provided some data from a Nikon commercial system. And so here is a cell that's sort of labeled for microtubules, and there's some clathrin pits and things labeled. This would be a wide field epifluorescence image, and then this would be the two-dimensional structural illumination reconstruction. So quite a bit of information gain, but I'm going to tell you immediately off the bat here, we're talking things on the level of 150 nanometers, right? 200 nanometers. But actually, all of that could be a lie too, depending on signal to noise. And so I'm going to talk a little bit about that. I don't have a super long presentation, so feel free to jump in with questions. Almost everything I'm going to tell you today, with the exception of the last sort of 5 minutes, is done by a postdoc from my lab who just recently moved on, Peter Brown, and who has a background in quantum gas microscopy and got interested in this. So there's some commonalities in terms of patterning lasers. So here's that same image. And you know, what I want to give on the first part here is what just a very brief background on what's going on here, and then I want to talk about maybe how do you deal with lower signal levels and get rid of those artifacts that you've run into, or at least our ideas. So I really like SIM because it's physics driven, which means we're going to pattern the light going in, and we're going to use something we know about the microscope to extract extra resolution information. There's a debate in our community whether or not to call this a super resolution technique, or do we just call it an extended resolution technique. And the reason I really like it is very few sample preparation requirements, right? So actually we just had this conversation about fluorophore physics sort of determines the STED beam. Here we can do this with pretty much any fluorophore, right? And so as long as it's fluorescence imaging, SIM works. And that's one thing I want to really emphasize is that there have been some confusion in the literature that this also worked with things like phase imaging or scattering imaging. That is incorrect. This is 100% determinant on the idea that you're imaging incoherent light. And so you need to be imaging fluorophores for the structural illumination ideas to work. And so, you know, why do this? So actually, this is great. Here's a tetrahymena. So this is imaged in my lab on a home-built system. On all the slides here, I'm going to give you what the sample is, what the fluorophore is, the excitation, and then the sort of specs of the system. So if you're curious, you can look on the bottom. So here's sort of standard 2D SIM, just Z stack through, so not 3D SIM. And then you can also do a little bit of post-processing to clean it up. And so these are the basal bodies. And so you can see even with just sort of standard image processing techniques in 2D SIM, we can go from a pretty blurry image of these areas where the cilia attach to the body, so this is where the cilia actually go into the tetrahymena, into something where we can see it. And so if we look at, you know, what this is, here we have that wide field image. Again, this would just be epifluorescence. And then here's a two-color version of that same sample with the basal bodies in cyan now and the cilia in orange. And again, this is 2D SIM, just taking Z stacks. You can improve the 3D resolution even more by doing 3D SIM, but it brings in other complications. And so I'm not going to focus too much on 3D SIM today. So this is pretty nice, right? This comes from basically manipulating the light that's going into the sample and running an algorithm to extract the information that comes out. With colleagues at UT Southwestern, particularly my colleague Radu Moldovan, we've been spending a lot of time thinking about how to adapt light sheet imaging to structural illumination. And so here is actually, this is a 3D projection that's color-coded. So here you can see 0, and here's 6.2 microns. And the point I want to image here is this is mitochondria that we're imaging at a rate of 1.2 hertz and actually doing that in 3D. So this is using this method that we've all been working on called single objective light sheet microscopy. And so we spend a lot of time working on this. We have a setup in my lab, there's a setup in Radu's lab, and so we've been doing it this way in 3D, and we've also, in his lab, we have not done this bit, adapted it to a special type of imaging that they've called projection imaging, where now you can actually boost imaging the whole cell in sort of a max projection type way at about 3 Hertz. So we're really pushing the speeds in these ideas. What often pops up though is people see these beautiful movies and then they go and try and do sim on their own and they get all sorts of artifacts inside of it, right? So I'm showing you data that's sort of selected, but the real trick here with sim, and I'm only going to go over 2D sim today, is that what I'm doing is I'm going to bring the laser beam into the back of my microscope objective, and then I'm going to interfere it with itself to generate this stripe pattern at the sample plane. And so when I do this and I overlay it with another structure, I get what's called a moire pattern. You may have seen one of these when you have 2 screens on each other. It turns out that there's high frequency information contained in this that I'm going to try and recover. And so, this movie is always problematic. And so here is a test target. So here we can define a resolution, right? And so what this test target is it starts with 290 nanometers separated in the middle all the way down to 0. And I hope everyone can see, maybe it shows up on this projector, sometimes the stripes are pretty clear and other times the stripes are not clear. So what's changing here? The orientation of this pattern is changing. And so actually, depending on the orientation of the pattern, I'm getting more or less information out of this sample. And the reason that's happening is, actually, I have an orientation in my sample as well. So if I try and pattern, with a pattern that's facing up on these, I'm going to get the most amount of information. Whereas if my pattern's facing this way, I'm lined up with the lines and I might not modulate it very much. And so you can think about this like looking through window blinds, right? If you're looking at something that's aligned through the window blind, I see it, where if it's vertical, I'm not going to see it very well. And so what ends up happening is people have done all sorts of strategies on trying to make these patterns. I would say that there's commercial implementations of all of these nowadays and you can buy them all. But in the end, how well that pattern shows up in your sample and how the signal to noise is in those patterns is ultimately going to determine the quality of the reconstruction. So that's a big difference between what we were just hearing about with STED and what we're hearing about here. Instead, that determination is on the photophysics and how you're doing the STED beam. Here, there is a computational step that requires us to somehow deal with noise. And that's really what bites the getting the nice data that I showed you in the end out of it. So again, there's tons of approaches on this. I'm sort of showing you the baseline entry here with this, the 2D sim, just so we don't get into these crazy geometric orientations that only exist in labs like mine, where physicists spend hours staring at small laser beams. All right. So this is the only equation I'm going to show. It actually doesn't really matter that much, other than to say you have to carefully control the polarization of the light. So if the light is polarized the wrong way, I actually don't get stripes. And so you have to be really careful about how you shoot these laser beams in. And so just to give you an idea of what that is, if I'm polarized correctly, I get a lot of contrast between the stripes when the two laser beams meet at the sample. If they're polarized incorrectly, I don't get very much contrast. And so if you have a setup and it seems like it's not working well, this is a great place to start. Is it actually aligned correctly? And so what do people do to build these things? So you can use a diffraction grating, and you can shine your laser beam through there and you manually move it. So the old Nikon setups do this, and it takes forever, right? So it moves, it rotates. Modern setups use spatial light modulators, and so these are faster, but they're still not that fast. My group works on a special type of spatial light modulator called a digital micromirror device. It's actually what's in this projector. It has good properties and bad properties. The good property is we can modulate it at 30 kilohertz. The bad property is it doesn't play well with more than one wavelength of light, so we've spent a ton of time working on how to use this thing. The idea is it's a bunch of tiny mirrors. The mirrors can tilt one direction or the other, and so you can change what's coming out. Here in the projector, it's using LEDs, and so there's not problems with interference. When you shine a laser off of it, you get all sorts of crazy diffraction patterns. And so what that looks like is this. Here's a, not laser safety necessarily approved movie from our lab. And so at the sample, there's that, but this is all the patterns that come off the device on the wall. And somehow you have to select the ones that you want out of it. So we have spent way more time than we care to do about this. Peter published a paper in 2021 that actually won the runner-up for best paper over 3 years in Biomedical Optics Express on how do you use these things for these types of experiments. Other people have adapted our work, and so the idea here is people have put it in head-mounted microscopes to do optical sectioning sim. We just wrote a paper with actually the originator of sim, Rainer Heinzmann, where they showed how to make a bolt-on unit to shove on the back of a commercial microscope that you can do 2-color sim with. And so they actually have like a rail and printing system where you actually just get all the stuff printed, drop all the lenses in, and it should immediately work. And so this just came out in Communications and Engineering like 2 weeks ago. So Reiners was great to work with because he's the one who originally had this idea along with Mats Gustafsson. And so, you know, it's easy to pattern light nowadays and it works and you can buy these things. And so what I'm showing you here on the bottom, this is mitochondria labeled with PK Mito Orange. So this is now the dye of everybody's choice. I just saw it in all the STED ones. So this is an ultra bright mitochondrial dye. Down here is the white box. And hopefully it's showing on the projector. Can everyone see the stripes maybe that they're moving through there? All right, so this is a relatively high signal to noise area. Now I'm going to turn on the movie for the yellow area. See a couple, but not that many. So the question is, when I reconstruct this, if I run the same algorithm that tries to unmix that high frequency information from both, I'm actually going to get a better reconstruction than I get up here, and I'm going to start to get some artifacts in the reconstruction. And so just to remind everyone where resolution comes from, it comes from this idea that microscopes only collect certain frequencies. That's all I'm really going to say. Too much about the image formation process. You have an object. That object, when light interacts with it, or if you have fluorophores, gives off light. Smaller structures are higher spatial frequency. And so eventually there are frequencies that are not captured by the microscope. The thing in incoherent imaging is that the microscope is worse at transmitting those higher frequencies, and so they're attenuated. So that means the SIM pattern is even worse in the sense that it's mixing the high frequency information in with the pattern, so now everything is at this very low signal level. And so when we go from our object to our image plane, there's all the physics in the microscope that's going to affect the way the image is formed. And so people can go over the resolution limit, you can measure point spread functions, you can do all these things till you're blue in the face. And none of it really matters for this method, because what's going to happen is if I start from an extremely bright signal and I have no noise in my detector, it's always going to work. But that is not reality. The reality is cameras are noisy, fluorophores are not perfect, and so if I now take these objects and I corrupt them using a simulated microscope, this is a pretty good microscope, and this is using a state-of-the-art camera, then this is, I would have this type of signal. Most normal cameras are going to look like this, and if I look at the histogram, my signal is sort of mixed in with the noise. And so the thing I want you to think about with SIM is it's this overall sort of modulation contrast. It's the contrast that matters for the reconstruction, right, is what we really care about. We still know the objects are there, but what we've seen in the image due to the physics of the microscope and the physics of the detector is that it's corrupted by all these processes. Okay. So the way the classic reconstruction algorithm goes that I mentioned a few times is you have an object, I know I illuminate it by a pattern, and then I know I blur it with my microscope. So I have some model of what I think the microscope's doing. So how do we process them? We actually do it in Fourier space, and so the idea is you take a Fourier transform of this. When you have your patterns, you're actually going to see information in the Fourier transform that's due to the patterns plus the original signal, and then we have to somehow unmix those. And in the process of unmixing those, the hope is we separate out the high frequency information from the low frequency information. I'm going to shortchange this explanation a bit because I'm going to tell you about a real space approach in a second, so I'm not going to spend too much time on this. The point I want to make about this is that the high frequency information is at the edge of the Fourier transform because the farther I go out in the circle here, the higher the spatial frequency, so this would be the lowest spatial frequency in the system. And remember, this is where the microscope is bad at transmitting information. So the thing I want is out where the microscope is not doing a good job. And so when I have noisy signals and I'm corrupted by noise, I have a harder time in disassembling the SIM image and putting it back into the real image because it's signal to noise dependent. Okay, so I know that's not great in terms of like a too in-depth explanation. What I want to really emphasize out of this is there's a whole bunch of parameters you have to estimate to get this right. So you have to estimate like what were the frequency of my SIM peaks? What was going on in the noise, which is this power spectral, right? What phases did I apply to shift my pattern? Right? What is my point spread function? There's all sorts of phonological signal to noise estimates. And there's a whole host of literature about how to best estimate these and how to do a good job. And people have been writing protocols papers since before I started in this field and pretty much as soon as the first SIM papers came out on when you see an artifact, what do you think that failure was in the computational reconstruction process, right? And usually the answer is take a better picture, but sometimes you can fix the algorithm a little bit. And so I really like this Nature Protocols paper from 2017 where they actually go through each type of artifact and sort of tell you where they think it came from, right? So you can see here if your sample has an RI mismatch, you get this ghosting up above it because your patterns are sort of deflected and they're not showing up where they think they're supposed to. All right, so it's 2025. Sort of AI is taking over, right? Deep learning is taking over. And so there's a mass of deep learning papers about improving these reconstructions. So their pitch is, don't worry about the parameters. We're going to take care of it by training a model. The results are impressive. The problem in our hands is that we have not been able to replicate a single one of these in a lab that's pretty technical and does a lot of machine learning on our own. And I don't know what the answer is for this, Will. I will say often authors are not very responsive, and I'm not picking on anybody. These results are stunning, and I love them. But in our hands, it's been hard to replicate. And so we were really interested in how do we remove this pain point of estimating the parameters, but how do we also make it sort of applicable to people that are working in this field where they don't need to retrain a deep learning model and struggle with this all very much. And so that's what I'm going to tell you about for the last bit here. But before I jump into kind of a little more of a rant on deep learning, are there any questions about the image formation model or anything I can clear up before I tell you kind of our approach to get rid of this parameter estimation problem?
So for the PSF, are you determining that empirically or are you using synthetic models?
So we measure it.
Okay.
I would say that it depends on, so my opinion is for most systems it's best to measure it. There's some debate, people like to measure the simmed PSF and then take it. In reality, the PSF that's being applied is the actual PSF of the system, not the sim PSF, right? So the thing that's in the image formation model is the actual PSF of the microscope. So we tend to use that. There's a lot of work about starting with that as a starting point and then modifying it to take care of any aberrations. That is not one of these deep learning models, and so the best one of those is called Hi-Fi Sim, and in fact that's an incredibly successful reconstruction technique, but it still suffers from all the other parameter estimation issues. So they're modifying the PSF basically to try and account for those irregularities to get rid of.
It does go a long way to removing them.
I agree with you, it does, and I'm going to show you results from their algorithm compared to what we were kind of working on. I think it's great. Hands down would recommend that as a standard sim reconstruction one. So that paper is fantastic. That method's easy to use. Their software is easy to use. All right. So, there's a lot of promise here. I really like this paper. David Hoffman, who's actually at 10x now, he was at Genelia, wrote this with some colleagues. If we train this thing on what we think our blurry images look like to get a cell, we can do that. But if I now reapply it to a cat, right, I'm going to start repredicting these things on the spot. And the problem with this is that there's no kind of physics built in at this point, right? It's a pattern matching problem. And there's other papers that have shown you can train on squares and then you can repredict and you can get out triangles or circles. The idea here is let's build some physics in. We know something about the microscope. In fact, this question you just had means we know something, right? And so there's a huge amount of ongoing efforts. My group has worked on doing GPU accelerated so we can do it in real time. We've worked on using irregular patterns like one would get if you're trying to use sort of nanostructures to generate the patterns instead of spatial light modulators. And so this is some of the work we did last year. And what I'm going to tell you about today is some work that we just published. It came out a couple months ago with colleagues of mine on doing this physically motivated optimization based. So we're not going to do it in Fourier space. We're going to do it in real space. It's going to have some computation that looks like de-learning, but it's going to be running a physics model. So what do? We take a guess at what we think the underlying sample is. So you can just start with your wide field image. So you take all your sim images and you average them together. You get a wide field image. That would look like this. Here's that PK mito stain. You can see I don't have great images inside the mitochondria. I know my PSF, which we need to calibrate. I know something about my patterns. And then I know something about my camera noise model. You don't have to calibrate your camera to do this. We can use the average parameters from your camera. So if you have the spec sheet, it'll work. Right? So you know something about the gain. How many electrons do I expect per photon? I know something about the offset, what's the noise floor, and I know something about the standard deviation in the noise. Any good camera manufacturer will supply this to you. So I take all this data, and this was all done both by Peter and Ayush Sarab, who was a postdoc with my colleague Steve Presse. So this is joint work between all of us. And what I do is I'm going to run that physics model I just told you about. But what I'm going to do this time is I'm going to include all of the noise in the model. So I know photons follow what's called a Poisson distribution. I know the camera typically follows a Gaussian noise readout distribution. So I'm going to simulate what I think the sim image would look like given an underlying set of the sample. So I'm going to guess what the sample might look like on a very small grid and then simulate for it. It's a guess, right? So when I make a guess, I need a way to compare that to what I think it is. So we do this in what's called the Bayesian paradigm. I'm not going to go through too much probability statistics. The idea is I get a probability of what happens, right? So you know the probability you flip a coin is heads or tails, that's 50%. We use the physics to ask how likely is what we predicted compared to what we actually saw. So it's called a likelihood, is what that's called in the math terms. So we calculate the model, we then calculate the likelihood, and we ask if our new guess, so maybe let's change the distribution of fluorophores a little bit, does better, keep it. If it doesn't do it, get rid of it and try again. So this is what's called a Monte Carlo simulation. And so the idea here is we're going to flip coins a bunch of times for where we think the fluorophores are, and what's going to happen is we're going to go from this wide field image to this reconstructed image. And so a movie might be a better representation of this. So here's the final result, here's my wide field image, here's my optimized image, and what's going to happen is we start at the guess, random guess, and then we start optimizing. And so what's happening here is every new proposal is being compared to what physics we think is going on and asked is it more likely or not. And so all of the same tools that are used to train a deep learning algorithm are being used to run the mathematics under here. But what it's being compared to is not training data you generated, but it's being compared to the physics of the microscope, right? And the fact that we have the sim images to compare to. So ultimately, what are we asking? How likely is it that the underlying distribution produced the raw data? We're going the other way, right? We're saying we're going to guess at the raw data, simulate the way the microscope works, and say, does that match the actual data we recorded using the realistic noise parameters? So this is called inverse modeling. So I often tell my students that the main thing they're going to learn when they leave, and what we never teach them in their classes, is this inverse approach for physics nowadays, right? How do I figure out what the most likely sample was given my data, right? What we often teach physicists to do is like solve Newton's equations, right? Go forward. So again, I'm still using this PK Mito die for all of this. This is all still our setup. And so the real thing that really is the proof in the pudding here, you know, is can we turn the signal down and still recover the same structure for this? So what do? We did a set of experiments where we imaged the same set of cells with that PK Mito dye again. It's very photostable, really recommend it. This is PK Mito red in this case. And then we generated an average number of photons per pixel. So on the right here, I'm going to show you that. So here, blazing bright, hundreds of photons per pixel, 800 is the average. What are we going to do? We just turn the laser down a little bit. So here, what I've got in the split here is what it would be if the data was scaled to this, if this was the max. And then here's the actual data scaled to display it on like a 595% scale. So here there's only 86 photons per pixel. You see we're order of magnitude less than we would normally run at. And then down here we're in tens of photons per pixel. So can we do this without getting all those artifacts, right? Because remember, the more noise we have, the lower the signal, the worse the microscope is at transmitting the information, the worse the artifacts. So here's the high reconstruction, just to give you an idea. Here's the hi-fi sim in the middle. So actually we use the hi-fi Wiener sim to do it here. Here's another optimization algorithm, which is called Fista sim, which is kind of hanging around in the literature. And then here's our result. Here's a line cut across this bit of the mitochondria. So you can see even at 532 nanometer excitation, we're sort of resolving crystase slightly, right? We're getting close there. But we're at least sort of doing better than the wide field image. Usually if someone told me they wanted to run at 16 photons per pixel, I would tell them this isn't going to work. And so here's the result. And so basically we're a little noisier, but actually if I flip between these, we actually have not lost that much detail, and we haven't started to generate these kind of classic hatch artifacts that are generated in the standard sim image. So in the paper, there's a whole host of other validations that go with this, but to me, this is the most striking example that at this kind of extremely low photon count, we're able to recover sort of pretty high matching the line cut, right, with some loss of information. But now we're running in a photon regime where we can image this cell for hours. We're barely putting light into it. And so this really opens SIM up into a regime where it's been difficult to work in before because the computational reconstruction doesn't work. The other nice thing about doing these optimization ones is we can actually tell you how certain we are in our guess. So we actually can put error bars on our sample. and tell you what are the areas of high confidence and what are the low confidence. So for those line pairs, right, when it's just one line pair, we have pretty high, sort of confidence in the areas outside it, but we're not very confident inside of it because we're not sure where we should put the fluorophores. Over here, where we're well separated, our error bars are really small, and then in the middle here, you can see we actually have some larger error bars on them. So this is something you don't get out of deep learning, you don't get out of the standard one, it's these confidence intervals. And this actually can be done across the sample to ask what it is. So nothing's free, there's never no free lunch, you should never even believe anybody who tells you that. What's the cost, right? So it doesn't require retraining, it doesn't hallucinate, it provides uncertainty estimates, but it's expensive computationally. So the point right now is that this is the type of thing you're not going to run, on a quick session at the microscope, right? Where you really want to use this is when you run through all of so here's microtubules, here's colleagues' turf sim data. This is another home built setup from Ohio, from the Corral lab. So we have sort of the wide field image, the Bayesian sim, the Wiener sim, and then they had tried the deep learning one. And if we zoom in here, you can actually start to see some kind of weird things happening in the deep learning, and if you come up and look afterwards, there's this cross-hatching pattern, which is like a classic one, which isn't supposed to be there. And so they asked us if we could kind of validate what we thought the sample was supposed to look like. And so where this algorithm really starts to come into its own is when you have this extremely low signal to noise, and you need to recover something like this. If you're in the high signal to noise, I want to point out, there's not much difference between the classical reconstruction method and running this optimization method, right? So a good place to start with people, and where we recommend, is like run a high signal to noise experiment so you know what it should look like, and then if you do need to record some low signal to noise one, then running this optimization algorithm, it might be worth the hours it's going to take to run. We're actively working to speed it up. There's sort of details and things that are tricky there that have to do with computing across multiple GPUs and all these things, and I'm hopeful it'll all work out in the end. But really, the idea here is when you have challenging data in sim, the problem is the noise. And the noise leads to poor reconstructions. And dealing with the noise in the reconstruction algorithm instead of guessing at what the noise parameters are is one way around this problem, right? I'm not telling you it's the only way, it's a way that we've recently introduced that we think is pretty exciting. All right, so to conclude, I really like sim. I think it's an incredibly powerful method that gets you this extended resolution, and it's helpful for a lot of things. If you want to count sort of the nice example we saw across a synapse, right, this is not the method. Right? We're not below 100 nanometers. But if you need to improve contrast and you need to improve resolution in that 100 nanometer range, it's a really flexible imaging method that works really well, works across all wavelengths. Of course, the lower the sort of bluer you are, the better the resolution. The hardware is it's incredibly stable, fast, and commercially available nowadays. There's bespoke ones like my lab does that go blazing fast, but you can get these things easy. People are putting out plans to build them on your own. In my opinion, the problem here is always just the reconstruction that people run into. Background, noise, all these things. And so this physically driven reconstruction really alleviates some of the problems, and our hope is to keep pushing this on the computation time so that we're closer to what it would be to kind of running the standard algorithm. One thing people will say about deep learning is, oh, it's so fast. It's only fast after it's trained. So one thing you need to think about is that if you need to retrain your data to not have these artifacts, our approach is actually faster.